Good morning, everybody. I always trick you into biochemistry when I start my talk, so we will start with biochemistry again. There are a few parents who are new to our meetings, so I want to start with sugarology. And this is a beautiful science because actually we use colors and forms to be able to communicate. And as you see, the different sugars all have their symptoms or symbols, because when you talk about, for example, galactose, which is milk sugar, it will be a yellow circle, or mannose, we call cranberry sugar, it's a green circle. So when you actually see a sugar chain on a protein, then you can see how is it built, and is it fully built or truncated or missing. So CDG is a very complex biochemical process, and as you see the little schematics on the top, it's like a step-by-step build of a lipid arm catching a sugar, and the next sugar is getting on top, and every different step could be a different disorder. So that's how we are reaching actually almost 150 different types of CDGs. Generally, we talk about two major groups, type 1 CDGs and type 2 CDGs. In type 1 CDG, the problem is that you're missing glycans. There are less glycans on your proteins. And on type, so that's type 1 CDG. And type 2 CDG, you do have your beautiful sugar antennas, but unfortunately, they are truncated. They are not fully finished. But you have enough antennas, just not completely ready to rumble. So this little, very short biochemistry is followed by our amazing dreams and projects together in the oncoming years, because we are so delighted that we were we received this grant in October last year, which we call Frontiers in Congenital Disorders of Glycosylation. And our consortium is, well, at least 70% from the principal investigators from the team are on this little picture. What we want to change, and what made us go for this grant, is that there are many, many new CDG types, and there are many undiscovered. But even when we know that somebody has CDG, sometimes we are unable to prove which specific type it is. There is no reliable diagnostics, and there is no reliable follow-up marker in many of the CDGs. There is no cure for most CDG types. CDG is an orphan disease, and expertise is centralized and scarce. And patients are frequently not equal partners in care. These were the emotional reasons we requested the grant, and our vision in the grant is to provide the best integrated clinical care for CDG in the nation. So from a patient's perspective, the services delivered are consistent and coordinated. Our mission is multifold. We want to improve clinical symptoms, improve quality of life and life expectancy in CDG. We want to share knowledge in CDG. We want to learn a lot about CDG and share it with all of you. And we need to develop a lot of new tools to diagnose the disease, but also to follow it, because we want to come up with 1,000 clinical trials, and we want to be sure that those clinical trials are safe and we are helping our patients. So this grant was only possible because the patients' associations are supporting us. They were helping us to write the grant. I'm so proud of this. I think we would have never get a grant like this without their help. And you see that we actually, you know, my background, I'm European, but not just that I have this personal connection to European patients and other patients, not just the US patients, but the European Patient Association is so much part of our community that they actually helped us with the grant as well. So that's pretty good because that's how we spread the news and get even more strong. So right now the consortium sites are here, and you see that all these sites will be a little, hopefully close to you, that you can participate in the different studies. You see a little color code. The reason for that is that Alabama and Sanford Burnham is not recruiting for clinical activities. They are our scientists, but many of the other centers have dual roles, not just clinical, but also scientific. And I also wanted to emphasize that Lynn Wolf joined us also, and Bill Gall also joined us and shares data with us, so we can actually get even more powerful to have all these sites united. And that's where we are right now, and we want to grow further and have as many centers as possible included. So finishing this introduction, the research focus is to define natural history, validate patient-reported outcomes, and share knowledge on congenital disorders of glycosylation. So that's the natural history study. The second very specific focus is biomarker discovery. And the third is to improve clinical outcome and develop new therapies. So I wanted to show you a couple of examples for what we are working right now and how we want to grow further. So first of all, just a little introduction to the natural history studies in the US. So in medicine, a natural history study is to follow a group of people over time who have a specific medical condition or disease and collect health information over time to understand how the medical condition or disease develops and give insight into how it might be treated. We need to understand the natural variation and the spectrum of symptoms in order to design a therapy and then know what am I changing by applying that new therapy. So that's one of our major goals. And I just want to give you a sneak peek where we are. So in this natural history study, we want to recruit additional 100 patients in between our different consortium sites. And I just want to give you an example. So for example, at Mayo, we have 61 patients currently who are coming for regular follow-up. We received the grant last end of October, and since then we recruited 26 patients, which is going pretty well, I think, and we would like to recruit them as soon as possible to have enough time to collect the data. So you see that it's sort of biased because it's happening at Mayo, but that's because a thing called central IRB, when we have our ethical approval, we have to share it with all the sites, and all the sites should agree that that's the ethical approval we need for collecting data. It's very sensitive and very strict. So it slows down the other sites. But Seattle Children's and CHOP are very close to being able to recruit, and the other sites are getting ready as well. It's interesting, in my 26 recruited patients, the youngest one was 10-month-old, and the oldest is 65. So it's really, I think, what we were talking about questions about getting to adulthood and hormonal abnormalities and what we don't know about that also got excited right away that this is something we specifically should collect prospectively within our cohort. So let me show you just one case from our 26 patients, because I think this case sort of moved many of our families within CDG care. So one finding, which is not a common finding in PMM2 CDG, is liver fibrosis. We had a patient who had so severe liver fibrosis that she got on the transplantation list. And I have to emphasize, the reason for transplantation was not for CDG. It was because of liver failure. So look at her poor big belly. She was really suffering from extreme liver dysfunction, ascites, and she was unable to sit, and her quality of life was pretty low. We did the liver transplantation at Mayo, and what we expected that, of course, the liver makes most of the glycans, so probably the transferrin will be normal, liver functions will be normal, hormones get normal, coagulation gets normal. So we were very optimistic, but actually we got so much more. Actually, what happened is the patient didn't have hypoglycemic episodes anymore, didn't have diarrhea anymore, which she has been suffering from since very early age. She started to use a sippy cup. She never used a sippy cup in her life. And again, these are little tiny things, but these are huge giant steps in quality of life, right? So the question comes like, is this because of just the liver transplantation, or is this because we were restoring glycosylation and it goes all over your body, it goes to your guts, it goes to your different tissues, and maybe some of the new glycans go also to your brain. So I'm not advertising for liver transplant in PMM2 CDG, but we want to use the knowledge we gather from this beautiful Lucy to learn more about these developmental therapies and how far restoring glycosylation could affect quality of life. So that was just a sneak peek from our upcoming natural history studies, but I also want to share with you the glycomine natural history study. There are many overlaps, and I don't want to go into too many details, just that glycomine increased the number of patients we could recruit, and the more European sites will also join, and more US sites probably will also join. And for that study, we actually have 70 patients already recruited. So that's also a gold mine of data on prospective clinical data. Also want to mention the CERCOR study, which is a retrospective data review study. They focus on a few disorder types, and their goal is to have FDA approval for nutritional sugars. So they are collecting this data to have enough data to go to the FDA and request FDA approval for sugar supplements in CDG. So let's switch to therapy development, and I'm hoping I didn't use too much time introducing you to the new natural history study, but let's start with a very simple example. So if you have PMM2 CDG, like how would you correct this defect? Look at your sugars. So it's fructose 6-phosphate, mannose 6-phosphate. Remember, this was the cranberry sugar. You have to make this change between the two forms of the molecule. And here's your enzyme, which is not working well. And the consequence is what I showed you before, that you're missing an antenna. So how would you treat this disorder? The easiest way would be to send in all those phosphate molecules in a way that it would get into the liver, somehow locked in a little lipid bubble, so probably it would be taken up by the cell and integrated into the sugar chains. And the idea is that that's how you could restore glycosylation. So this trial is not ready to rumble yet. This is hypothesis. Glycoman is working on safety trials, and they just showed this, slide yesterday about their timeline. So right now, they're just in the natural history phase of data collection, and they try to have their safety studies done this year. And if everything is going well, then it might be a phase 2 trial next year. So this would be some sort of sugar therapy, but in a very tricky way. So what are dietary therapies? This is not dietary therapy. That would be probably an infusion therapy frequently given IV, right? But there are other therapies which we are using every day in CDG to improve glycosylation. So different sugars would get built into the sugar chain, and if you miss them, you try to give more and hope that the glycan chain will get restored. So the indication is either not sufficient or not, the sugar is not present in a sufficient amount in the sugar chain, or somehow adding the extra sugar would increase the activity of sugar chain production. So when we are giving sugar supplement therapies, it's not for the calories. We are giving building blocks or metabolic tricks which could help glycosylation. So a very classic treatment method is in 1B, CDG 1B. You see the same graph, just the block is here before the PMM2. And so what you can give a whole lot of mannose, cranberry sugar, which would get activated in the body and would restore glycosylation. So the same approach can be used in a couple of other CDGs. So the three sugars which we use the most frequently in different CDGs are mannose, galactose, and fucose. So I'm not saying there is enough evidence to call all these sugar therapies a treatment, but there is already some evidence, and there are clinical trials out there. So this is maybe a very simplified evidence graph, how much we know, how much these sugars help our patients, our children with different CDGs. And you see that if you go down on this chart, for example, fucose therapy has been only trialed in 6 or 7 patients altogether at different centers. So we cannot really say that it's actually a therapy. There are many risks of sugar therapy. You have to be sure that you not overdose your sugars, because you can have side effects of that. Families have difficulties to get these sugar supplements because they are not prescribed. Sometimes they get it from Amazon and you don't know what's in there. So it's really important that we actually use reliable sources, and that's why Cerecor is also trying to help us to have FDA-approved sugar types. Okay, so I think we can conclude that there are still a lot of challenges for dietary therapies, and these are the 3 clinical trials currently ongoing for fucose and galactose, and they are mostly at Mayo Clinic and CHOP. And you can see in the graphs next to this little patient that all of the therapeutic indexes are going down, which is a good thing. You see also this therapeutic index in PGM1 going down. It means that severity score is improving, and this is our most recent therapy trial in SSC 35A2 CDG. You see that the improvement is less dramatic, but most patients reacted well. So I would like to finish up with 3 very brief announcements about upcoming clinical trials, which could be important for your child as well. Based on Perlara's work in vitro, we started the first individual, so it's a single patient IND therapeutic trial in PMM2 CDG with this aldose reductase inhibitor, which seems to increase the enzyme activity in patients who have residual enzyme activity. And based on this single patient IND, we hopefully can start a clinical trial if we do find that this drug is safe in our children. The second drug I would like to mention is acetazolamide, and Mercedes Serrano is sitting in the audience. This is a diuretics which we know that has effect on periodic movement disorders. We have been using, actually at Mayo Clinic, Mark Pettison has been using acetazolamide in ataxia patients. And there is a great clinical trial which was done in Catalonia, which showed efficacy of this drug in the clinical outcome of CDGs. So this is the trial we are planning here in the US. It's a placebo-controlled, randomized, double-blinded study for several years to be sure that this drug is safe and to find the best dose and see whether it really significantly improves the clinical outcome in CDG. And here is a preliminary graph showing that in 6-month period, how much improvement can be in the writing pattern in a child who is taking this medication. We are not sure that all the patients will react the same way. We still have to learn a lot about this. And the last trial I would like to announce is Abilify in Angli-1. There is already in vitro data and the case report data on the efficacy of this molecule in Angli-1 patients, and we would like to recruit patients for a safety trial, and we already started the IRB, submitted an IRB for this at Mayo. And I would like to thank you for being patient with me, always going over time. But I hope I showed you the most important highlights you need to know to that. And you need to know that we are really, our team is all here and they're excited, and we cannot wait to move forward and show you that we can do much better. Thank you.